Printer is a machine for printing text or pictures. Keep this concept in mind. Well, genes are also imprinted on the chromosomes. That encodes different protein. During gametogenesis, several hundred genes receive genomic imprinting. That normally ensures later expression of only one of their two inherited alleles. In contradiction of Mendel's laws, mutations of such genes can cause dramatically different disorders, depending on whether they were inherited from the mother or the father. The conditions are of particular importance in this regard are Angelman syndrome and Prader-Willi syndrome. But before moving into detail, let's understand the basic concept. Maternal and parental. Chromosome 15 has ubiquitin protein ligase E3A imprinted on it. This encodes for E6 associated protein. This protein tags other proteins with ubiquitin. Then this ubiquitinated protein are eaten by protease. Lack of ubiquitin protein ligase E3A cause lacking of E6 associated protein, and hold of this process is inhibited. Moving forward, maternal chromosome 15 and as well as parental chromosome 15 has Angelman site and Prader-Willi site. Moving into details, as we have discussed that this is maternal chromosome 15 and this is parental chromosome 15. For normal brain function, one copy of a lone sequence called the small nuclear ribonucleoprotein polypeptide N must be transcribed and produce small nucleolar RNA, and one copy of the sequence. Ubiquitin protein ligase E3A must be expressed as the protein ubiquitin ligase, Angelman site, inactivated by methylation, and Prader-Willi site, inactivated by methylation. Let's understand it in more details. Prader-Willi syndrome is caused by lack of expression of normal alleles of small nucleolar RNA. Angelman syndrome is caused by lack of normal alleles for ubiquitin protein ligase E3A product and ubiquitin ligase. In order to memorize it, remember that in case of Angelman syndrome, father's chromosome or parental chromosome is silent, and in case of Prader-Willi syndrome, mother's chromosome or maternal chromosome is silent. Let's memorize these using mnemonics. Angelman syndrome and Prader-Willi syndrome. Moving forward, this is father's or paternal chromosome 15, and this is mother's or maternal chromosome 15. So these are paternally and maternally imprinted chromosomes in normal brain cells. The blocks represent structural genes, and the vertical arrows their expression. Differential patterns of imprinting in paternal and maternal chromosomes are indicated by blue and pink coloration, established by appropriate activities of the differentially activated imprinting control center. Now let's discuss the causation of Angelman syndrome and Prader-Willi syndrome one by one. In case of Angelman syndrome, we know that in Angelman syndrome, parental chromosome is silent. So, first cause of Angelman syndrome could be maternal deletion. Second cause could be that if identical copies of one chromosome are inherited from father, called as uniparental paternal isodisomy, which has Angelman site, inactivated by methylation. Moving to third cause, we know that in Angelman syndrome, parental chromosome is silent. So, third cause could be maternal imprinting error. Moving to fourth cause, in Angelman syndrome, parental chromosome is silent. So, fourth cause could be the point mutation in ubiquitin protein ligase E3A. In Angelman syndrome, parental chromosome is silent. So, there may be point mutation in ubiquitin protein ligase E3A. Now, let's discuss the features of Angelman syndrome. Angelman syndrome exhibits epilepsy, severe neurodevelopmental delay, uncoordinated movements, and compulsive laughter, happy puppet attitude with jerky movement. Now, let's discuss the causation of Prader-Willi syndrome. We know that in Prader-Willi syndrome, maternal chromosome is silent. So, first cause could be paternal deletion. Moving to second cause of Prader-Willi syndrome. If identical copies of one chromosome are inherited from mother, called as uniparental maternal isodisomy, which has Prader-Willi site, inactivated by methylation. Moving to third cause of Prader-Willi syndrome, we know that in Prader-Willi syndrome, maternal chromosome is silent. So, third cause could be paternal imprinting error. Now, let's discuss the features of Prader-Willi syndrome. Obesity, inverted V-shaped upper lips, and small hands and feet. Short stature. Hypotonia. Hypotonia means decreased muscle tone. It can be a condition on its own called benign congenital hypotonia, or it can be indicative of another problem, where there is progressive loss of muscle tone, such as muscular dystrophy or cerebral palsy. It is usually detected during infancy. Compulsive eating. Hypogonadism. Hypogonadism occurs when the body's sex glands produce little or no hormones. In men, these glands are the testes. In women, these glands are the ovaries. And mild to moderate neurodevelopmental delay. Now let's discuss maintenance methylase. Maintenance methyltransferases add methylation to DNA when one strand is already methylated. These work throughout the life of the organism to maintain the methylation pattern that had been established. This is DNA sequence with methylated CpG and non-methylated CpG, C for cytosine, and G for guanine. DNA polymerase acts and produced hemi-methylated DNAs with one old strand and one new strand. Now symmetrical methylation is restored by a maintenance methylase enzyme that scans the new DNA strand for hemi-methylated CpGs. So, duplicate copies of original DNA are produced. So, this was all. Thank you for learning. Join us again.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.